Hi, I'm Ola Landgren. I'm professor of medicine and chief of the myeloma program at Memorial Sloan Kettering Cancer Center in New York City. Today we are in New York City and we just completed the fourth meeting, Advances in MRD Testing in Multiple Myeloma. With me here today, I have Dr. Dikran Kasanjan, investigator at the FDA and the National Cancer Institute, Professor Faith Davis from University of Arkansas Medical Sciences, and Professor Jens Hillengas from the University of Heidelberg in Germany. Thank you very much for coming, all of you.
Thank you for having us.
I think it was a very exciting meeting today. So, as I just said, it's the fourth time we had this meeting. I felt this meeting just keeps on getting better and better. This meeting, in my opinion, was the best we have ever had. What do you guys felt? You don't have to agree with that.
I thought it was a great meeting, and what I liked most was that compared to last year, it was better, but you could see, I saw a sequential improvement in the technology, and I think that's part of the reason why it was such a great meeting. And I think the whole atmosphere, I really love this meeting every year because the atmosphere and the camaraderie between everyone involved is always great.
What did you think, Faith?
I agree. I mean, it's a really unique meeting. We've got all of the different stakeholders in the same room talking about how we can really make MRD, what we can do with it, how we can improve patient care with it, and how we can maybe improve drug approvals. And so having everybody in the room, and as you say, seeing what progress has been made from last year. Last year, the result was we should write a white paper about where we were, and we've done that. And now the next question is what we're going to do next. And the new technologies and everything was really encouraging.
What do you think, Jens?
Yeah, like very much that we had a lot of discussion. It was the talks themselves were kind of short, but they came with a lot of discussion, a lot of questions, and a lot of also answers. And I liked it so that it was really a work meeting. It wasn't just like showing off or just telling what you think, but really discussion and going steps forward was really nice. Yeah,
I think it really takes a good group of people that work hard and are smart to kind of pull this off. So we had these short presentations about a lot of unpublished data with new technologies and new approaches, and people were willing to share. And then, as you just said, we spent most of the time talking about all these things for the new things. But also, if we go back and look through the structure of the meeting, we started off actually by looking at data that's already completed from clinical trials. So we saw MRD negativity is observed in 2/3 of the experimental arm for the French multicenter study and in 1/3 of the control arm patients. And that translates into the same progression-free survival. However patients were treated, they had the same progression-free survival if you use 10 to -6 negativity. And then we saw very similar results even in relapsed myeloma, that the quarter of the patients obtained MRD negativity. And I presented a short update on the smoldering trial we did at the NCI, also showing very high rates of MRD that was sustained almost 4 years out. So what's your thinking, Jens, when you see these results?
I mean, it's so exciting because it came so fast. When several years ago we were happy when we achieved the CR, and now we have to step even further and develop more sensitive techniques to see really what is still there from the disease. And I think I dare to say this has to be the first step going to a cure of this disease, because if there's nothing measurable there, it's a very good sign. If it's really zero, we do not know that yet, but I think we're on a really great way, and it's exciting to see what happened in the last few years, and it's really great to be a myeloma doctor at the moment.
Do you think that these observations, Faith, are going to impact the design of new clinical trials?
Without a doubt, I think one of the discussions today was about what technology to use. And as you say, the studies that have been reported so far have suggested that 10 to the -6 is the level that we need to aim for. There's a number of other groups that are using slightly different technologies and are looking at 10 to the -5. And so there was lots of robust studies and discussions about whether it should be 10 to the -5 or 10 to the -6 as the measurement. But you can see that everybody is now thinking about, we should be having that into clinical trials, but also what clinical trials can we perform? What can we do with that? Should we be aiming to try and get as deep as we can for response? Should we be thinking about stopping or adding in treatments if patients don't achieve that kind of level? And so there was lots of discussion about what the next steps should potentially be.
So after this session on the clinical trial update, that's just brand new, that was, again, unpublished data even in this session. It just happens as we speak, all these new things. We had a second session focusing on how do you, as an investigator, use MRD in your clinic? And you were one of the speakers on that, Faith. But I'm going to ask you, Deco, what did you hear from that presentation? Did you see a message like that was consistent, or did you see differences across the different centers?
I think the message was that, again, MRD, in the clinic is still really important. But the message I got was kind of, a lot of that is to gain more experience in understanding MRD negativity in patients. But at this point, there isn't quite enough data to sort of, off of a clinical trial, to guide management of patients based on MRD by itself.
I think one of the presenters, Dr. Sergio Giralt from Memorial Sloan Kettering, he said that I'm going to show a couple of patients to illustrate my clinic. And if I recall his presentation, he said that the patient had certain characteristics, and then the patient was MRD positive, and it was a 1-hour appointment.
Right.
And then they did things, they took action, and then they treated again, and that was a new 1-hour appointment. And I think that was kind of reflective of the fact that there are a lot of things that we need to kind of work out, as you're indicating, Faith.
Yeah, I think there are lots of questions we need to answer. As you say, if you go through treatment and you're MRD positive, do you need more treatment to make you MRD negative? Or indeed, if you're MRD negative and then suddenly become MRD positive, what does that mean? And we don't really know the answers to those questions. And I think, as you say, what one of the other speakers in that session was saying was, I guess, some of the practicalities of trying to do MRD in centres, particularly maybe in the community and so on. I'm really lucky. I work in a big centre. We have an incredible setup to be able to do bone marrows and imaging. But if you're in a smaller centre, how do you get access to those tests, and how do you ensure, if you do have access to the tests, that they're done properly and that you know how the quality control has been performed? And so I think that was an interesting discussion that went on.
So the next session after that focused really on all these new technologies. So we took it from the right out of the presses, new clinical trial data, and we took it into the clinic, where there is still some work to be done. And then we moved into the future with the new assays. So we covered a lot of different assays. We had the sequencing-based assays. There were free circulating DNA. There were single-cell sequencing approaches. There were some proteomic approaches. There was some update on new flow approaches. And then, Jens, you spoke at the imaging part of this presentation. So can you update us a little bit on your perspectives on the imaging? Where is imaging going?
Actually, it was before the talk, I became more and more afraid that it's really relevant until then, because the people were speaking about measuring MRD in peripheral blood, and I thought, okay, maybe the focal lesions that are seen in imaging are not that relevant anymore. But I think it's still relevant, because at the moment the best sample we can get is bone marrow still, and so if we perform a biopsy of the pelvis, which we do usually, we still have to consider that there are accumulations of plasma cells, residual focal lesions in other parts of the body. So it's really relevant to have an at least impression, maybe not with a sensitivity, of course not with the sensitivity of a bone marrow biopsy, but still you get an impression of the whole body, and you can see, okay, if you have eradicated most of the cells in the pelvis, you should be sure that it's done the same way elsewhere. And it was very interesting to discuss if MRD negativity on 10 to the minus 6 may be also like a sign of a negativity in imaging as well. And we do not know that yet, but it will be exciting to look into those data. And I think, Faith, you are doing it, and we are doing it in a collaborative approach to see how the imaging and the MRD from peripheral blood and also from bone marrow correlate.
So what I heard from these discussions was that people believe in 10 to minus 6 over 10 to minus 5, but until we get there, we shall continue to collect 10 to minus 5, but we have to keep track of if it's 10 to minus 5 or higher, so we have that in our record. So with the current guideline, 10 to minus 5 is the lowest level. And if you look at the data that was presented, I think it seems that the 10 to minus 6 can deliver better progression-free survival across the different studies, but we still have more work to do so we can expand the cohorts. Is that a fair statement?
I think so, yeah, because there's lots of subtleties that we need to work out. So, and it may be that we end up with different criteria for different things that we want to look at. Because certainly we have, it's a small data set, but the timing of doing the MRD in a high-risk patient to give you the best answer moving forward may be different to that in a low-risk patient. And we know that some patients will take a long time to get to an MRD status, whereas other patients may achieve it quicker. And so there's some final subtleties in there, not necessarily for maybe using this as a clinical endpoint for a study, but maybe more for using it on a day-to-day basis when we're seeing patients in the clinic. So I think that we probably all agreed that having the higher sensitivity was very important, but at the moment, because of those subtleties, we need to have as much data as we can, so we should maybe include everybody.
Yeah, I agree with that. I agree with that. So the next session after that was a session where the FDA graciously contributed. You contributed to this DECO in your capacity both as an NCI and an FDA investigator. So you gave an overview of where the agency is right now and looking forward. To me, it's just amazing to see how very progressive and dynamic the agency is. You are very scientifically driven. You even talked about setting up assays at the FDA to further understand the nuts and bolts of these technologies.
I mean, I think it's really important for us in the agency, you know, and for, as I mentioned during my talk, about half of the clinical reviewers are actually medical oncologists who still see patients at least once a week. And so I think that's been important to actually be able to be progressive and without seeing patients, without understanding the new technologies, without really getting into some of the nuts and bolts, which you can only do with your own experience. It would be hard to sort of understand how certain things in the field may go forward. So it kind of makes us kind of more in context and more real, to say.
I really enjoyed all your presentations. I think you did a fantastic presentation. Your colleague, Nicole Gormley, did a very nice presentation. And I think, at least I felt she was looking at me when she was giving her talk, and she said, "You have to do this now," she said. And I looked at her. I felt, probably all of us felt like that almost the agency tell us you have to move forward and work with MRD and make it a regulatory endpoint. Also, Gideon Blumenthal, I think, was a fantastic contribution. He agreed to be on this lunch interview, and we had this discussion with him in an open fashion and asked a lot of questions, and I think that was just fantastic.
Absolutely.
So we wrapped up the meeting content-wise by talking about the need for guidelines, and we all agree that guidelines are needed. We got the first guideline last year, and it's very easy always to criticize guidelines because many times guidelines are written based on assumptions and not always data. And if you know the data, then you can find all the holes. But I think we were all in agreement that it's too early to update them. They need to be updated, but it is too early.
Yeah, I think so. So I think the conclusion was we're expecting such a lot of data to come through in the next year that will hopefully clarify many of these questions that we're asking, that potentially saving it for a year and then doing an update then would be a really good opportunity.
What was your take on these sessions, Jens?
Yeah, I also think that I think it should be also from a European perspective, I would say it should be a global approach because there's so many data coming up, as you mentioned, from France, from Germany as well. We are also doing MRD in our trials. And I think if we have those data, which are not, we don't have to wait 5 or 10 years to get them. I think it will be in a few years, and then we have a really huge data set in different settings, different trials, different treatment paths. And so I think that will be a great opportunity. And I think what you should start, as you mentioned, we should start now to start to collect, to set up the infrastructure, to really then have the baseline and to work from that and to collect those data sets and put them together and yeah, come up with a really even more reliable guideline. Yeah.
I agree with that. So if I were to summarize the meeting here today, I think we had a fantastic day with review of existing data looking into the future. And we ended the meeting by concluding, all of us, that we now have a task force going forward. We are working together with academia, with pharma, with the NCI, with patient groups and MMRF and other organizations that work to support the myeloma research field. And we are going to work together to pool data sets that come from both academic and from drug company studies. We're going to work with the FDA. We're going to set up meetings with FDA shortly to replicate what has been done in other diseases with the aim to make MRD a regulatory endpoint for drug approval. I think this is huge, and I think we agreed, so we better go home and work after we're done here today, because we agreed that we're going to do this in 1 year. So with that, I think we conclude this discussion. Thank you very much for coming.
Thank you very much.